Inside the human cell, a structure called a nucleus. Inside the nucleus contain a chromosomes. Each chromosomes contain a DNA molecule with histone protein. So we have these two chromosomes. First one of one chromatid, the second is two chromatid, and the first one contains one DNA molecule with histone protein, and the second one contains two DNA molecule with histone protein. DNA stands for deoxyribonucleic acid, and it's found inside the nucleus, which codes for genetic information. And we see in this picture that the nucleus is extracted from the cell, and the chromosomes from a nucleus, and DNA from chromosomes. So DNA is found inside the nucleus. And we can see in this diagram that DNA is composed of two strands twisted around each other, so DNA has a double helical structure. The two strands connect with each other by hydrogen bonds and is made up of phosphoric acid, deoxyribose sugar, nitrogenous bases, and there are four types of nitrogenous bases: adenine A, thymine T, cytosine C, guanine G. And A, which is adenine, pair with thymine by a double hydrogen bond, and cytosine pair with guanine by a triple hydrogen bond. DNA replication takes place in the interphase, and chromosomes of one chromatid become chromosomes of two chromatids to be able to make mitosis. And interphase has three phases: G1, S, and G2. DNA replication is the biological process of producing two identical replicates of DNA from one original DNA molecule. During the G1 phase, only cellular growth takes place, and the chromosomes are filaments made up of one chromatid. So, chromosomes at G1 is made up of one chromatid and its filaments, and it has centromere, one DNA molecule, and histone protein. And because it has one DNA molecule, so it has two strands. During S phase, a synthesis phase, a DNA replication takes place. DNA is opened by helicase enzyme at numerous places along the strands, so the two strands are separated by helicase enzyme, creating Y shape as we see in this picture, and it's called replication fork. And then the DNA polymerase enzyme adds new nitrogenous bases found free in the nucleus, where each old strand acts as a template strand for the formation of a new strand. So, chromosomes at S phase is made up of one chromatid and centromere, one DNA molecule, and histone protein. And the area that makes replication, it's named as eye of replication or replication bubble. And in this video, we see how DNA replication takes place and how helicase enzyme functioned and how it separated the two strands, plus how polymerase enzyme functioned by adding new nitrogenous bases. As we see, each daughter DNA actually consists of one old DNA and one newly synthesized strand. And the cases we see in this pack, ensuring our explanation of DNA replication. So, during G2 phase, the final phase, two DNA daughter molecules is formed. Each consists of one old strand and one new strand. So, during the G2 phase, the chromosomes are filaments of two chromatids. It's made up of two DNA molecules centromere with histone protein, and the two DNA molecules, each one contains one original strand and one new strand, as we see in this drawing. DNA replication is semi-conservative, meaning that each strand in the DNA double helix acts as a template for the synthesis of a new complementary strand. This process takes us from one starting molecule to two daughter molecules with each newly formed double helix containing one new and one old strand. I'm Raghad Rashid. I will leave you with this short video. Enjoy.